All right, this is the beginning of the second lecture exam material covered by exam 2 this semester. We're going to talk about microbial metabolism. Before we get into specifics here, though, this picture is showing us a very cool form of metabolism, and these are mushrooms, forest mushrooms. Don't remember the actual species name, but they are bioluminescing, so they actually glow in the dark. Very cool. Anyway, before we get started, I want to say a couple of words about the term metabolism. That term is often used incorrectly, so I want to go through and make sure we're all on the same page on what that term means and give a couple more specific terms. The term metabolism encompasses all of the chemical reactions and physical processes that occur in a cell. So all reactions that happen, all of the enzymatic reactions, all of that stuff in its entirety is called metabolism. Now we can be more specific in calling things either catabolism or anabolism. Catabolism are the degradation pathways. The catabolic pathways take large macromolecules and break them into smaller molecules, and those processes release energy. So when you ingest your breakfast and then you break it down and get energy from it, those are the catabolic pathways. Those are the degradation pathways. They release energy. On the right-hand side of this diagram in blue are the anabolic pathways. That's anabolism. Anabolism takes small molecules and uses them to build up large macromolecules. So these are the biosynthetic pathways. They generally require an energy input in order to function. So metabolism encompasses both catabolism, the degradation pathways, as well as anabolism, the biosynthetic pathways. A couple of words about energy and what energy is and types of reactions here. It turns out that all molecules have a type of potential energy called chemical energy in their bonds. So if you think back to physics, I think they cover this in chemistry as well, there are two different types of energy: kinetic energy, which is energy of movement, and then there is potential energy, energy of position or structure. Chemical energy is a type of potential energy. Chemical energy is the energy that is in a molecule's chemical bonds. So it's an energy of structure, right? How those atoms are arranged. Because of how those atoms are arranged into bonds, those bonds do store some potential energy, and we call that chemical energy. More specifically, it's called Gibbs free energy. Gibbs free energy is the energy that a molecule has because of its chemical bonds. When a molecule enters into a reaction, right, if it's the substrate being turned into product, when that molecule enters a reaction, it either absorbs energy from the environment or it releases energy into the environment. So there is a change in the energy of that molecule. So if you think about something like glucose getting broken down into carbon dioxide, glucose is a large molecule with several chemical bonds in it. It has a high Gibbs free energy. As it breaks down into carbon dioxide. Carbon dioxide is a very simple molecule without such large chemical bonds, so it has less Gibbs free energy. So in that case, you go from high energy substrate to low energy product. You are releasing energy into the environment. We're going to go through that example one more time in a few slides. So the energy change between before and after the reaction is called delta G. The Greek letter delta, looks like a triangle there. The Greek letter delta always means change in science. So when you see delta G, you know we're talking about the change in Gibbs free energy. And we look at the delta G, and it's actually calculated by taking the G of the products and subtracting the G of the reactants. And that's how we calculate delta G. So delta G, or the change in Gibbs free energy, is the G energy of the products, subtract the G energy of the substrates, and that gives us the delta G. Reactions that have a negative delta G release energy into the environment. Reactions that have a positive delta G absorb energy from the environment. Right? Let's look at some examples here. You can term reactions as being endergonic, or we'll see in a moment exergonic. This is an example of an endergonic reaction. And what you'll notice here is we have carbon dioxide, right, simple molecule, low energy in that substrate, with some waters, also low energy, getting turned into glucose and releasing oxygen. This is what photosynthesis does. It takes low energy carbon dioxide and water and eventually produces high energy product, that is glucose. So if we look here at the free energy of carbon dioxide is very, very low. It's a very simple structure. So you see here there's low energy in the substrates, but there is high energy in the products. Product here is glucose, a much more complex molecule. So we know that energy has to be put in order to produce this high energy product. The delta G, remember delta G is equal to the energy of the product subtracted by the energy of the substrate. So if you take a big number and subtract a small number, you're going to get a positive value. So the delta G is positive. In fact, this particular reaction requires 686 kilocalories per mole of energy to be inputted. So in other words, this is an example of an endergonic reaction. The products have more energy than the substrates. There is a positive delta G here. This reaction absorbs energy from the environment. Sometimes they call this a non-spontaneous reaction because you have to put energy in to have this reaction occur. So it's not spontaneous. Let's look at an example of the other side here, an exergonic reaction. If we take the exact same reaction but flip it around, now our substrate is glucose, high energy in our substrate, being turned into carbon dioxide, very simple structure, low energy in carbon dioxide. So we're going from a high energy substrate to a low energy product. That means energy is being released, right? Because we're going from high energy to low energy. How much energy is being released is given in delta G. Now remember, delta G is the energy of the product, subtract the energy of the substrate. So if you have a low number and you subtract a high number, you're going to get a negative value. So exergonic reactions, reactions that release energy, always have a negative delta G. In this case, negative 686 kilocalories per mole. So this is an example of an exergonic reaction from a high energy substrate to a low energy product. It has a negative delta G. It releases energy into the environment. You could say that this is a spontaneous reaction. Spontaneous indicates that you don't have to put any energy in for this reaction to occur. It will occur naturally. It will want to occur without any outside energy input. Now, of course, the cell has to do both endergonic reactions and exergonic reactions. Exergonic reactions like this one release energy. So that seems very obvious how that cell why the cell would want that reaction to occur. But of course, the cell also has to do endergonic reactions. The cell also has to be able to build up large energy products. So what the cell does is what's called energy coupling. Energy coupling is when the cell uses an exergonic process to drive an endergonic process. So energy coupling is when the cell uses an exergonic process to drive an endergonic process. For example, let's look at an example here of energy coupling. Here's a reaction that the cell would need to do. The cell is going to take glutamic acid and ammonia and put them together in order to produce glutamine. Glutamine is one of the 20 amino acids that every cell must have in order to make protein, so all cells have to make glutamine. But just looking at this reaction, you should be able to tell that glutamine is a very complex structure and therefore will have a higher G, higher energy, than either glutamic acid or ammonia. In other words, this is an endergonic reaction. Energy will have to be put in order to synthesize glutamine. It's not spontaneous. The delta G is positive, right? Positive delta G, energy absorbing. How much energy does it cost? Well, it costs 3.4 kilocalories per mole for this reaction to go. So the question is, where does the cell get that energy from? Where does the cell get the energy to drive this endergonic process? And the answer is energy coupling. The cell is going to use an exergonic reaction to drive that endergonic reaction. The most common exergonic reaction to use is the breakdown of ATP. Here we see ATP. When it gets broken down into ADP, this is a highly exergonic process. ADP is a more simplistic molecule than ATP, and therefore you do get energy out when you break it down into ADP. It is an exergonic reaction or a spontaneous reaction. How much energy do you get from the breakdown of ATP? Turns out that you get 7.3 kilocalories per mole. That's the energy you get when you break down an ATP molecule. Delta G is negative because it's an exergonic reaction, indicating higher energy in the substrate than there was in the product. So from here it works pretty much like checkbook math. It costs the cell 3.4 kilocalories per mole to make glutamine, so it's going to break down one ATP and get 7.3 kilocalories per mole out of that. So put this together and what you have left over is 3.9 kilocalories per mole. This is a very common way for the cell to get the energy to drive these endergonic reactions. You're going to couple that reaction to something that's exergonic, and if you get enough energy from the exergonic process, you can run that endergonic process. And of course, in live organisms, the leftover here, got 7.3 from ATP, right? You use 3.4 of it to make glutamine, you have 3.9 left over. Generally in the cell, that's just given off as heat, right? That's just kind of wasted out into the atmosphere. So energy coupling happens continuously in the cell. It allows the cell to do these endergonic reactions, the biosynthetic reactions that are absolutely essential for the cell to build up its macromolecules. Now, what exergonic reaction is most common? This is the most common exergonic reaction to use, the breakdown of ATP. Breakdown of ATP is used all the time for energy coupling. It's an essential process. It's an essential reaction to the cell. Let's look at why energy is so effectively stored in this ATP molecule. First off, here you see the structure of ATP. ATP stands for adenosine triphosphate. The structure here of ATP, this together, this is adenine, right? Adenine plus ribose, a sugar. It's a nitrogenous base called adenine plus ribose, which is a sugar. Together, this makes adenosine. Right? So all of this together is adenosine. And what you'll see is here is adenosine with 3 phosphate groups. Phosphate group 1, phosphate group 2, phosphate group 3. That's why they call it adenosine triphosphate. If you only have 2 phosphate groups, then we call it adenosine diphosphate. If you only have 1 phosphate group, then we call it adenosine monophosphate. So that's all that ATP means, is it's this adenosine molecule with 3 phosphate groups attached to it. Now, think back to chemistry. In chemistry, you learn that different functional groups have different charges on them, sometimes positive, sometimes negative. If you think back, what kind of a charge does the phosphate group have on it? Phosphate groups are negatively charged. So if you look at the structure here of adenosine triphosphate, you have adenosine and then 3 negatively charged phosphate groups attached one to another. Do 3 negatively charged phosphate groups want to be attached to each other? Of course they do not, because they're negatively charged, they are repelled from one another. So they often will show these phosphate to phosphate bonds. They're showing you here. They'll often show these phosphate to phosphate bonds. They call them high energy phosphate bonds. That's kind of a silly term because there's nothing special about the bond other than the fact that it's between phosphate groups that happen to be negatively charged. Therefore, this bond is just dying to break. This bond wants to break. This phosphate group wants to be released. It does not want to be next to the other phosphate groups that are also negatively charged. This phosphate group also wants to be broken. It also wants to be released. So there is a lot of energy in the phosphate to phosphate bonds because those phosphate to phosphate bonds are holding negatively charged groups together. So when one of these groups does fly off, when ATP loses a phosphate group and becomes ADP, it does release energy, and it releases, if you'll remember from this slide, 7.3 kilocalories per mole. Right? ATP being broken down to ADP releases 7.3 kilocalories per mole. So that's a good energy amount that's given off from that reaction. And if you look at the structure of ADP, so if you lop off this phosphate group and you have ADP, is there still energy left in the structure of ADP? And the answer is yes, because adenosine diphosphate has two phosphate groups attached to one another still two negative groups attached together. There is still energy in this bond, so it does still release energy if you were to take off another phosphate group. Not as much as when you break down ATP, but you do still have some energy coming off. Okay. So where you get the energy here, let me move this out of your way, is removal of that terminal phosphate group releases energy because those phosphate to phosphate bonds hold a lot of energy in the fact that they are negatively charged. So what the cell's going to do is go back and forth between ADP and ATP. ATP is going to hold a lot of energy and release it when you break it down into ADP. So the cell uses this regeneration and then breakdown of ATP in order to drive reactions in the cell. So for example, you have ATP. When the cell needs energy for the biosynthetic processes, right, those endergonic reactions, the cell is going to break down that ATP into ADP and release energy that can then be used to drive endergonic processes and endergonic reactions, like the construction of glutamine. Here, ADP is then going to get turned back into ATP. How does the cell get the energy for that? Well, it gets the energy for that from catabolism, the exergonic pathways. So you eat your lunch, and your cells break down those molecules in order to get the energy to make ATP. And then the ATP is used in order for your cells to do the work they need to do. And then you eat dinner, and that energy is used to make some more ATP out of the ADP, and then that ATP is broken down in order to drive more cellular work. So that's regeneration of ADP and ATP over and over and over again. In fact, this goes incredibly quickly in the cell. The cell is constantly regenerating its ATP and then breaking it down. About 10 million ATP molecules are consumed and then regenerated per cell per second. So 10 million ATP molecules going through this cycle per second per cell. Unbelievable rate. Constant regeneration. A working muscle cell will completely go through all of its ATP once per minute and then have to regenerate. So very, very common reaction. And again, the reason is because then the ATP breakdown can be used in order to drive some of these endergonic processes. So it's a great way to store and release, store and release, store and release energy as it's needed by the cell. So because ATP is so vital, you're going to see that it plays a major role in connecting catabolic and anabolic pathways. Remember, the catabolic pathways are the breakdown pathways, taking these large molecules that we ingest and breaking them down. The energy that we get from catabolism is used to make ATP from ADP. Then the anabolic pathways, the biosynthetic pathways, will take that ATP, turn it back to ADP, and use the energy released in order to make those macromolecules. So ATP is really the central molecule that's constantly being used and then regenerated in order to allow these reactions to occur. Now, we're going to talk both about catabolism and anabolism, but first I want to talk about catabolism and the ways to synthesize ATP. How does the cell do that? Turns out there are 3 main catabolic strategies for making ATP. Different cells use different strategies. Some cells can use multiple strategies. We're going to use this slide as an overview. We'll come back to it several times as we go through all of these different pathways. First here, aerobic respiration, then anaerobic respiration, and finally fermentation. So this slide, again, we're going to see it over and over again. It's kind of a big picture slide. So it's going to show you the pathways involved in each process, what's needed at the beginning, what's released at the end, and how much ATP you get from that strategy. So aerobic respiration first. Most organisms on the planet do aerobic respiration. We do aerobic respiration. Most microbes also do aerobic respiration. The basic pathway here for aerobic respiration is one that we've already seen here. This is the basic sort of chemical reaction of aerobic respiration, where you take 1 glucose and 6 oxygens and turn it into 6 carbon dioxides and 6 waters. That's the basic aerobic respiration pathway, right, or reaction, I guess, that encompasses all of that activity. We're going to see that there are 3 steps, if you will, to aerobic respiration. Glycolysis happens first. From glycolysis, then you enter into the tricarboxylic acid cycle, which is second, and from there you move into the electron transport system. So we're going to go through each step one at a time. Starting out here at the beginning with glycolysis. First, let's talk about location. Glycolysis happens in the cytoplasm of both prokes and eucs. So glycolysis occurs in the cytoplasm of both prokes and eucs. Now it turns out that glycolysis really has kind of 2 different phases here. What they call the energy investment phase, right at the beginning of the pathways. Right at the beginning of glycolysis, it does require 2 ATP in order to get the pathways moving. So you do actually use up 2 ATP during the early steps of glycolysis. But later on, you actually produce 4 ATP through the later steps of glycolysis. So if you do the math here, if you spent 2 ATP to start the pathway, but you got 4 out of the pathway, you're ahead by 2. Your overall yield of ATP here is 2. Glycolysis pathway takes glucose, which is this large 6-carbon sugar, and it breaks it down into pyruvate. Pyruvate are 3-carbon sugars, so therefore there will be 2 of them. Right? So glucose goes in, you get 2 pyruvates out. 4 ATP are made, 2 ATP are used up early, so you have 2 ATP is your yield. And you also get a molecule we call NADH. I just want you to have that in your notes for now. We'll go back and talk more about that in a little bit. But these NAD molecules go in and we get NADH out. 2 go in, 2 come out. So glycolysis is handy because it's 2 pyruvate, 2 ATP, 2 NADH, 2, 2, and 2. Now, I want you to know glycolysis from what's often called the black box approach. So you'll think of glycolysis as just being this set of reactions. You don't need to worry about what's happening inside of that box. I just want you to know what goes into the box of glycolysis and what comes out. So here you see glycolysis, glucose, a 6-carbon sugar goes in, 2 pyruvates, 3 carbon sugars come out, 2 ATP go in, 4 ATP come out, so you're netting 2, and 2 NADs go in, and they get turned into NADH during glycolysis. So your yield here is 2, 2, and 2. Now that's the level to which I want you to know glycolysis. I have included the next slide that kind of gives you an idea of what's going on in this box. So the next slide is really what's happening inside of this gray box here. What does glycolysis mean? It turns out that glycolysis actually encompasses a whole series of reactions with a whole bunch of enzymes all functioning together. So if we look here, glucose again goes into glycolysis. An enzyme called hexokinase turns that glucose into glucose 6-phosphate. Another enzyme called phosphoglucosylaisomerase takes that glucose 6-phosphate, turns it into fructose 6-phosphate. Notice that hexokinase required ATP for its reaction to occur. Phosphofructokinase also requires ATP. That's why you use up 2 in the early processes. Here, an enzyme called aldolase is splitting up this large 6-carbon structure into the 2 3-carbon structures. They then continue on, eventually becoming pyruvate through all of these steps. Notice the production of ATP along the way. Notice the change here from NAD into NADH along the way. Now again, I do not want you to know all of the enzymes and all of the intermediaries. What I want you to recognize, however, is that the term glycolysis is a whole bunch of enzymes all functioning together. That's the level what I want you to know. I want you to know what goes in and what comes out, the black box approach that I have here. But I wanted you to recognize that this glycolysis box actually is a whole bunch of different enzymes functioning together. All right, so we've gone through glycolysis. Pyruvates are coming out the other side here. Where do the pyruvates go? The pyruvates go to the next step of aerobic respiration. So we went through glycolysis here. The pyruvates are coming out and heading towards the tricarboxylic acid cycle. Now the tricarboxylic acid cycle has a couple of different names. You may have learned this before as the Krebs cycle. You may have learned this before as the citric acid cycle. All 3 of those names are the exact same process. I don't know why they have 3 different names for it. I'm using the same name that's in your book, but don't let it confuse you. If you've learned it as Krebs cycle before, it's the exact same thing, or if you've learned it as citric acid cycle, it's the same thing. All right, so what happens here? The pyruvate that came from glycolysis, the pyruvate is going to enter here into the tricarboxylic acid cycle. You can see the production of some NADH. You can see actually the production of some FADH2s as well. See some carbon dioxide getting released through the steps of the tricarboxylic acid cycle. If we look at yields here, what comes out of the tricarboxylic acid cycle? What comes out of the TCA is you get 1 ATP for every pyruvate that goes in. Here it's being made here. You get 4 NADH. Here's 3 here and 1 there. You get 1 FADH2 and you get 3 carbon dioxides. Here's 2 and there's the other. That's the yield per pyruvate, where pyruvate's coming in. What do you get out of it from the tricarboxylic acid cycle? However, remember that for every glucose that goes into glycolysis, you don't have just 1 pyruvate. Instead, you have 2 pyruvates. So if you think about the yield per glucose instead, everything happens twice. Right? So you get 2 circles of the tricarboxylic acid cycle. So therefore your numbers double. You have 2 ATP, 8 NADH, and 2 FADH2, and 6 carbon dioxides. Now, why am I making a big deal out of this? Most of the time when people talk about the catabolic pathways here, if they ask you about tricarboxylic acid cycle, they're generally asking about per glucose. However, I have seen some very tricky standardized test questions that ask you about things per pyruvate. So I wanted to make sure that you guys understand what the difference is and how to tell if this is per pyruvate or per glucose. If you're asking questions about how much ATP do you get from the tricarboxylic acid cycle per pyruvate, that's 1 turn of the cycle. But really, most of the time we calculate it per glucose because glucose goes into glycolysis and we count everything that happens from that point. Right? So 2 different numbers here, depending on if we're talking about per pyruvate or per glucose. While we're here, let's talk about location. Where does the tricarboxylic acid cycle occur? In a eukaryote, the tricarboxylic acid cycle occurs in the matrix of the mitochondria, which is the very inner part of the mitochondria. So in eucs, TCA happens in the mitochondrial matrix. In procs, it happens in the cytoplasm. Okay. So if you remember, glycolysis happens in the cytoplasm for both eucs and procs. TCA happens in the mitochondrial matrix of eucs, but in the cytoplasm of procs. Now, a moment ago, I showed you that glycolysis was actually a whole series of reactions that all happen together. The same is true for the carboxylic acid cycle. I want you to know it from this level that we just talked about, what's going in, what's coming out. But I do want you to be aware of the fact that the tricarboxylic acid cycle is actually a whole number of reactions, several enzymes, all functioning together in order to run this tricarboxylic acid cycle. Pyruvate comes in through a couple of steps, and then several enzymes will turn it into citrate, and then isocitrate, and alpha-ketoglutarate, and succinyl-CoA, and around it goes, slowly pulling off carbon dioxides, forming some NADH, 1 little ATP, and 1 FADH2 as it cycles around. So again, you don't need to know all of the enzymes and intermediaries. I only want you to know it from the black box approach, but I wanted to make sure that you understand that the tricarboxylic acid cycle is actually a whole series of enzymes working together to run this cycle. All right, so let's think about where we are so far. So far, if we look at aerobic respiration, we have encompassed both glycolysis and the tricarboxylic acid cycle, the first 2 steps of aerobic respiration. So far, glucose came in to glycolysis, pyruvates came out, right? If we're following the carbons, 6-carbon sugar getting formed into 2 3-carbon sugars. 2 ATP went in, 4 came out, so we yielded only 2 from glycolysis. 2 NADs went in and they became NADHs through glycolysis. Then, of course, we encounter the tricarboxylic acid cycle. The pyruvates went into the tricarboxylic acid cycle, and those carbons came out as carbon dioxide. Right? So glucose goes in to glycolysis, becomes pyruvate through glycolysis. Pyruvates go into the tricarboxylic acid cycle, and the carbons come out as carbon dioxide. Carbon dioxide, of course, is then exhaled and leaves the cell. There's very little energy in carbon dioxide. It's generally released. Tricarboxylic acid cycle also made 2 ATP during that process per glucose. You had 8 NADs coming in and being released as NADHs. You also had 2 FADs going in and being released as FADH2. So far, here's our tally. We have glucose going in, 6 carbon dioxides coming out. We have a total of 4 ATPs that have been made so far. 2 were made here in glycolysis, and 2 were made here in the tricarboxylic acid cycle. Now what we haven't talked about yet, but we're going to do this now, is these other molecules. This NADH that was made in glycolysis and the NADH that was made in the TCA and these FADH2s. Turns out that some energy has been trapped in this NADH and FADH2, and the NADH and FADH2 are going to move on to the next step, the electron transport system, in order to release that energy. Before we get into the electron transport system, however, I want to review a topic that I find many students did not completely understand or have forgotten about since they took chemistry. I want to talk about oxidation versus reduction reactions and energy that's associated with them. So oxidation, reduction reactions, redox reactions here, these are chemistry terms that I know you learned once upon a time, but let's do a quick refresher. The term oxidation means loss of electrons. Reduction is the gain of electrons. Now, I realize that that seems backwards in terminology. However, this is what the chemists call it, and it is their term, so we are going to use it as they have made it for us. I can tell you why they named it this way, though. They called reduction the gain of electrons because it's reducing the charge. Remember, electrons have a negative charge on them. So when something gains electrons, it's actually reducing its charge. That's why they have that term. Now, unfortunately, that doesn't really help us in biology because it still seems backwards, but all I can tell you is you just got to remember that reduction is the gain of electrons, while oxidation is the loss of electrons. Now, it can be difficult to identify in a reaction which way the electrons are going in a chemistry lab. However, in biology, it can be much easier. It turns out that because biological organisms always live in an aqueous environment, water is around, for that reason, electrons always travel with protons or hydrogen ions. So write in your notes, in biology, electrons always travel with an H, right? A hydrogen ion. So you can always tell what molecule has the electron because you can tell where the H's went. Let me show you an example. Here's a reaction. In this reaction, we're taking methane, CH4, right, plus oxygen here, and it's getting turned into carbon dioxide plus water. Now, if you follow here, we have methane getting turned into carbon dioxide. As he's changing, is he gaining or losing electrons? Well, hopefully you're realizing he is losing electrons. How do I know that he's losing electrons? Right? How do I know he's losing electrons? Because he's lost his H's. And electrons always travel with H's in biological reactions. So methane is becoming oxidized, turning into carbon dioxide. So this is an oxidation reaction. On the other side, oxygen is getting turned into water. Okay? So is oxygen gaining or losing electrons? He's gaining electrons. How do I know? Because the H's always travel with the electrons. So oxygen is gaining hydrogens, H's, in this reaction. Therefore, he is becoming reduced. He gained electrons. So oxidation reduction reactions tend to be easier in a biological setting because, again, we all live in an aqueous world and hydrogen ions will always travel with electrons when you're talking about biology. Now, this will not always work in a chemistry lab, but it will always work in a biological organism. So that's the good news and the bad news of redox reactions. So I would expect if I gave you a reaction like this, you would be able to identify who's being oxidized, who's being reduced in this reaction. Other ways to look at it. Water is the reduced form of oxygen. This is the oxidized form. Here you're seeing this reaction. This is oxidation. Carbon dioxide is the oxidized form. Methane is the reduced form. Well, it turns out that electrons carry a small amount of energy with them. So when something becomes reduced, a little bit of energy is being stored in that molecule. So for example, in glycolysis, NAD goes in, two NADs go in, they come out as NADHs. Are they getting oxidized or reduced during glycolysis? They're getting reduced during glycolysis because they go from NAD to NADH. So they're getting reduced. And in fact, the reduced form has the extra electrons, and those electrons carry a little bit of extra energy. So there is energy in the structure of NADH. Same thing's happening here. NAD is going into the tricarboxylic acid cycle. It's coming out as NADH. Therefore, the NAD has been reduced and is storing a little bit of energy. Same with the FAD to FADH2. FAD goes in, it comes out as FADH2. That's the reduced form that stores a little bit of extra energy in it. Let's look more closely at NAD versus NADH. NAD, by the way, stands for nicotinamide adenine dinucleotide. So you see nicotinamide here, nicotinamide adenine dinucleotide. Now the structure of NAD here is a very intense looking structure. Here's the structure of NAD, here's the structure of NADH, almost identical. The only difference here is whether or not that extra electron and hydrogen is here. Here's NAD, here's NADH. Right? That's the only difference. That's really the only important part of that structure for our use. So when something goes from NAD to NADH, that's reduction as you load on those extra electrons and hydrogen along with it. Right? So this is a higher energy form than the oxidized form of NAD. So what happens here is in glycolysis and TCA, they're reducing NAD to form NADH. Right? They're loading on those electrons. What's going to happen is NADH is going to deliver those electrons to the electron transport system and become NAD again. Then it will go back and get some more electrons from glycolysis and TCA. We're going to see that FAD and FADH2 work exactly the same way. FAD or FADH2 stands for flavin adenine dinucleotide. It's also a very intimidating structure with only one little part that really matters, and that's whether or not those extra electrons and hydrogens are there. And of course, think for yourself here, which of these two is the oxidized form, FAD or FADH2? Which is oxidized? And the answer is FAD is the oxidized form. FADH2 is the reduced form. Which one has more energy in it? FADH2 has more energy in it because it has the extra electrons and hydrogens. Alright, so our NADH and our FADH2 that were made here in glycolysis and TCA. Here we had NADH, right, made in glycolysis, NADH made in TCA, FADH2 made in TCA. Those are going to travel on to the electron transport system. Now this is a new slide that I just put together. I usually draw this on the chalkboard during lecture, so I'm trying to kind of do the same idea here by building a diagram. Hopefully this will work for you guys. This is the electron transport system and a process called chemiosmosis. I'll explain what that is in just a moment, but first I want to orient where we are in the cell. In a eukaryotic cell here, we are located in the mitochondria. This is the matrix, or the inside of the mitochondria. Remember, the mitochondria has two membranes. Here's its inner membrane that's folded. Here's its outer membrane. On this side would be the cytoplasm of the cell. All right, so just to orient you where we are, that's where we would be in a eukaryotic cell. Now it turns out that the electron transport system is mainly made up of a whole series of proteins that are located in the inner membrane of the mitochondria. So in blue here are our ETS proteins located in the inner mitochondrial membrane. Now what's going to happen is NADH is going to show up. Remember, NADH was formed during glycolysis in TCA. NADH is going to come up and he gets close to one of these proteins, and the proteins will pull off that electron, turn NADH back into NAD. NADH just got oxidized to be turned into NAD. That electron is now here on this first protein. So this first protein just got reduced. I'll back up and do it again. NADH comes up. NADH is the reduced form. Our first protein rips off NADH's extra electron. NADH just got oxidized into NAD. He lost his electron. Our first protein just got reduced. He gained that electron. Next, the second protein in the chain is going to pull that electron away from the first protein. Okay? So the electron is pulled to the second one. The second one just got reduced because he just got the electron. The first one just got oxidized because he just lost his electron. Now, the third protein is going to pull the electron away from the second protein here. So this guy just got reduced. This guy just got oxidized. Now this one's going to pull the electron away from that one. So this protein, so this one just got reduced. He gained the electron. That one just got oxidized. He lost the electron. Likewise, the last one here, he's going to pull the electron away. He just got reduced. That one just got oxidized. This is why they call it the electron transport system. The electrons come in on NADH, and they travel to the first protein, and then they get passed off down the chain of proteins, right, or transported. That's why it's called the electron transport system. So why do electrons move down the chain? Right? Electrons continue to move because each protein of the electron transport system has a higher electronegativity, is what it's called, than the last. In other words, each one is more electronegative than the protein before it. Electronegativity is an attraction for electrons, sort of like a magnetism for electrons. And it turns out that each of these proteins has a stronger attraction for electrons or electronegativity than the protein before it. That's why electrons travel. For example, if we lined up, this is what I usually do in a class scenario, if we lined up a whole bunch of sports stars, right, and we go from the weakest of the sports stars to the strongest of the sports stars, right, we line them all up and we give them all something that all sports stars want. My husband suggests steroids, but let's be a little bit more PC. How about Gatorade? All sports stars want Gatorade. So we're lining them all up here from the weakest of our sports stars at this side to the strongest of our sports stars on that side, and we give the first one something that they all want, like Gatorade. So here, our first protein, our first sports star maybe is a track and field guy. He has that Gatorade. Well, standing next to him is the Olympic swimmer, and the swimmer is stronger than the track and field guy, so the swimmer pulls the Gatorade away. Right? Well, standing next to our swimmer is our baseball player, and our baseball player is even stronger, so he's going to pull the Gatorade away from our swimmer. Right? Standing next to him is a basketball player. Basketball player is even stronger than our baseball player, and therefore the basketball player is going to pull that Gatorade away. Okay. Then we have our football player. Football player is even stronger. He's going to pull the Gatorade away from the basketball player. So in other words, they pass that Gatorade or electron from guy to guy, from protein to protein, because each one is stronger than the protein before it. So where does this go? At the very end of the chain, the strongest of all sports stars, the one and only, the sports god, if you will. Did I tell you I went to school in Washington State? The hockey player. The hockey player is the strongest of all sports stars. He's there at the end. He can pull Gatorade away from anyone, no matter who they are, because he's just a badass like that. So in our actual electron transport system, who is the hockey player? The hockey player that can pull electrons away from anybody is oxygen. Oxygen is extremely electronegative. He is one of the few molecules that can pull electrons away from just about anyone, and that's what's at the end of the electron transport system. Oxygen is there. Oxygen is going to take electrons away from our football player here, our last protein, and oxygen will become reduced as he gets those electrons, and the reduced form of oxygen is water. So in the electron transport system, right, electrons travel from NADH through the ETS proteins and eventually onto oxygen to form water. Oxygen is our final electron acceptor. He's at the end of the chain, pulling those electrons off the chain. This is why you have to have oxygen. You must have oxygen for your electron transport system to work. Without it, the electrons have nowhere to go and you stop electron transport. So it's absolutely vital to have oxygen as the final electron acceptor. That's the first part of ETS, is this movement of electrons from NADH eventually down the chain to oxygen. Now, FADH2, by the way, functions very similarly. It delivers electrons just the same way electrons travel down to oxygen. Now, this is all well and good, but we haven't actually made any energy this way. We've just moved the electrons down. Well, it turns out that as electrons move from one protein to another, the energy of the electrons moving is used to pump a proton across the inner membrane of the mitochondria. So as the electron passes, that proton gets pumped into this inner membrane space, the space between the two membranes. Electron moves again, and another proton gets pumped. Electron moves again, and another proton gets pumped. So as the electrons move, those protons, or hydrogen ions, are getting moved into the inner membrane. Eventually, as more and more electrons move, more and more NADH delivers electrons, you get a huge concentration difference of protons across the inner membrane, where you're going to have a whole bunch of hydrogen ions here and almost none on this side, right? And that happens because the electrons are moving and those protons are getting pumped. You have created the very definition of potential energy. Large concentration of one side of the membrane, small concentration on the other side of the membrane. Do those protons want to be highly concentrated and next to each other? Absolutely not, right? Absolutely not. They want to get away from one another, but they can't because they're trapped by these two membranes. This is called proton motive force. Proton motive force is the huge concentration difference of protons across the inner membrane of the mitochondria. It's a form of potential energy. You have all of them on one side. They would love to come to the other side. So if I back up and repeat myself here, the energy of electrons moving down the electron transport system is used to pump protons into this inner membrane space. Eventually, all of those protons build up a huge proton motive force, where you have a large concentration of protons on one side and low concentration on the other side. And of course, those protons are dying to get back to this side. They want to go down their concentration gradient. They want to get back into the matrix of the mitochondria. Well, there is one way in, and only one way. there is a tunnel through the inner membrane of the mitochondria. This tunnel is an enzyme called ATP synthase. And ATP synthase is the one and only way for protons to get back to the other side, back to the inside of the mitochondria. What happens is, as those protons go, and they want to go because of proton motive force, as the protons move through ATP synthase uses that energy to make ATP from ADP. So it's actually the proton motive force that's running the production of ATP. This side of the equation here, this side of our diagram, is called chemiosmosis. Proton motive force, and using the proton motive force for protons to come through ATP synthase and making ATP from that reaction is chemiosmosis. I'll come back to this in just a moment. All right, so it is actually proton motive force and this large concentration of protons that drives the production of ATP through the electron transport system and chemiosmosis. Here's a picture of what we sort of drew out together. I think it's nice to look at it in steps though, but now we'll go and look at the really nice picture that they have for us. Here we have the mitochondria, the two membranes of the mitochondria, the inner membrane, the outer membrane. This is the matrix of the mitochondria inside the mitochondria. This would be the cytoplasm outside the mitochondria. Here you see NADH. NADH is coming in, dropping off his electrons, becoming NAD. In other words, he's getting oxidized. Our first protein here just became reduced. He transfers those electrons to the second one. He transfers to the third. Get my arrow back here. To the fourth, to the fifth, down they go. And our final electron acceptor, of course, is oxygen. Oxygen gets reduced to form water. That's our final electron acceptor, absolutely essential for this process to occur. The energy of electrons moving is used to pump protons. The protons accumulate in this inner membrane space, and we call that concentration difference proton motive force. Proton motive force will drive the protons back through ATP synthase, and ATP synthase synthesizes ATP as they move through ATP synthase. So the second half here, chemiosmosis. Chemiosmosis is actually using the proton motive force in order to make ATP. As those protons come down through ATP synthase loads on a phosphate group to synthesize ATP. That's what chemiosmosis means. So how effective is this system? It turns out that 3 ATP are made for every 1 NADH that enters the electron transport system. So you get 3 ATP from 1 NADH. You get 2 ATP for 1 FADH2. So if you look back at our diagram here, for every 1 NADH that enters, you wind up getting 3 ATP. Turns out that FADH2 enters a little bit later in the process, and therefore for 1 FADH2 that enters, you only get 2 ATP out. That's the efficiency for those. Now, I've been talking about the electron transport system in the mitochondria of Eucs. Let's just talk briefly about where this is located in Prokes. Remember that Prokes, of course, don't have a mitochondria, but their enzymes, those electron transport proteins, must be in a membrane. The one and only membrane that Prokes have is the cell membrane. So indeed, that's where you see this activity occurring. Here's the cell membrane. Here are the electron transport system proteins, right? NADH would deliver its electrons. The electrons would travel down to oxygen. You have the pumping of the protons. The protons accumulate in the periplasm instead of in the space in the inner membrane space of the mitochondria. Other than that, it works the same. Protons build up proton motive force. They come down through ATP synthase and synthesize ATP. Really, the only difference is location. ETS and chemiosmosis occurs in the plasma membrane of prokaryotes instead of the mitochondrial inner membrane. So let's put all of these different parts of aerobic respiration together. Right? If we encompass both glycolysis and the tricarboxylic acid cycle together with the electron transport system and chemiosmosis. Remember, in glycolysis, we had glucose going in, 2 pyruvates coming out, 2 NADHs were made or coming out of glycolysis, 2 ATPs were made as well. The pyruvate then went into the tricarboxylic acid cycle. Tricarboxylic acid cycle for those 2 pyruvates released 2 ATPs. Also produced 8 NADHs, 2 early on, 6 later on, and 2 FADH2s. So if we add up here NADHs that are going into the electron transport system, 2 went in from glycolysis, right? 8 NADHs are going in from the tricarboxylic acid cycle. So that means a total of 10 NADHs are going into the electron transport system. So 10 NADHs going in. Remember that the cell gets 3 ATP for every 1 NADH. So since there are 10 going into the ETS, 10 times 3 is 30 ATP that's made. There are also 2 FADH2s that are going into the electron transport system. Remember that the electron transport system makes 2 ATP for every 1 FADH2, so that means 4 more. So that means that in electron transport and chemiosmosis, it's possible to make 34 ATP in that process. If we add that up, 2 ATP that came from glycolysis, 2 ATP that came from tricarboxylic acid cycle, 34 that come from the electron transport system. Huge number coming from the electron transport system. Add all up together, and that gives the possibility of 38 ATP that can be made for every 1 glucose that starts this process. So I would definitely expect that you would know how many ATP are made in aerobic respiration, but also how and where and which cycles make the most, those sorts of questions. So a total of 38 are possible. Now, not always do you get 38, but in theory, you could get as many as 38 ATP for every 1 glucose that enters into aerobic respiration and the 3 steps of aerobic respiration. So if we look back at our slide here that compares the different metabolic strategies, aerobic respiration, which is what we've been talking about. Most organisms on the planet do aerobic respiration, including most microbes. And they use glycolysis, they use TCA, and they use the electron transport system. Remember what goes in, glucose goes in, it gets broken down into carbon dioxide. Right? Oxygen goes in, and it's essential because it will get reduced into water. Now, a couple of questions I could ask you. Where does the carbon dioxide get released from? Carbon dioxide gets released from the tricarboxylic acid cycle. Which part of aerobic respiration requires oxygen? The electron transport system requires oxygen because it's the final electron acceptor. I could ask you, which portion of aerobic respiration makes the most ATP? The electron transport system and chemiosmosis make the most ATP. 34 of the 38 come from those processes. So the total possible here, ATP made per glucose in aerobic respiration is 38, a big number, lots of ATP made there. That's aerobic respiration, the most common form of metabolism. Let's talk about anaerobic respiration. Anaerobic respiration is only done by a few organisms. It's really only done by some prokaryotes. While we're here, I know that in physio, they're using terminology a little bit sloppily. I want to bring to your attention, sometimes they talk about anaerobic respiration human bodies, but human bodies do not do true anaerobic respiration. What they're talking about is a form of fermentation. They're kind of using that term incorrectly, so I don't want that to confuse you. True anaerobic respiration only happens with some prokaryotes. It's not something that humans can do. In fact, it's not something that any eucs can do at all, that anaerobic fermentation. All right, so the process here of anaerobic respiration includes glycolysis, TCA, and the electron transport system, pathways we're already familiar with. In fact, glycolysis happens exactly the same way as it did for aerobic rest. In fact, TCA happens exactly the way it happened in the aerobic rest. There is a slight change to the electron transport system. That's the only difference here. Other than that, it's identical. So what's going in and coming out is very similar here. Let's look at how it works. Anaerobic rest. Anaerobic rest uses glycolysis, TCA, and ETS. In fact, glycolysis and TCA are identical. The only difference between aerobic respiration and anaerobic respiration is the final electron acceptor of the ETS. Remember, the ETS of aerobic respiration, the final electron acceptor was oxygen that became water. Anaerobic rest uses something besides oxygen as the final electron acceptor. What's being used there? There are lots of possibilities. Generally, it's some sort of a salt, an inorganic salt that's being used. Nitrate, nitrite, sulfate. These are very common salts that procs can use as their final electron acceptor. There is variety there, but it would never be oxygen. Right? If there's oxygen at the end of the ETS, it's aerobic. If it's not oxygen, then it's anaerobic rest. How much ATP is being used here depends on the salt that's being used as the final electron acceptor, depends on the electronegativity of that salt. There is some variety, but generally it's about 1 to 2 dozen ATP that you get during anaerobic rest. Never as much as you'd get with oxygen. Remember, oxygen is a hockey player. Oxygen has an extremely strong affinity for electrons, so he can make a lot of ATP through that process. Anaerobic rest doesn't make as many. So if we compare anaerobic rest to aerobic rest, same pathways here, glycolysis, TCA, ETS, glucose is going in, carbon dioxide is coming out, because remember, glycolysis and TCA work the same. The only difference here is in the final electron acceptor. You have a salt as the final electron acceptor, and what you get is a reduced form of that salt because it accepts the electrons and becomes reduced. How much ATP you get does vary depending on the final electron acceptor that's used. About 12 or 2, 10 to 25, I think, are the possibilities here for ATP. Notice it's never as good as it would be with oxygen, again, because oxygen is such a strong electronegative molecule. All right. The last of the possible catabolic strategies that I want to talk about here is fermentation. Fermentation is done by some procs as well as some eucs. In fact, this is what your muscle cells do. Your muscle cells can do a form of fermentation as well as other organisms can as well. Fermentation is going to use glycolysis exactly the same way that we've seen glycolysis before. But after that, it's not going to do TCA and ETS. Instead, it's going to go through a different pathway called fermentation. So let's look at how this works. Fermentation strategy uses glycolysis and then this fermentation pathway. Glycolysis works exactly the same as glycolysis always works. You have glucose going in, getting broken down into 2 pyruvates. You have 2 NADs getting reduced to form NADH. And you have 2 ATPs being formed during glycolysis. Remember, the net yield was 2. So glycolysis works exactly the same way as it always works. You get 2 pyruvate, 2 ATP, 2 NADH. Okay. Now, the fermentation strategy, in fermentation, from that point, the fermentation process uses those pyruvates, and it also uses NADH to yield some kind of an alcohol or possibly an acid. So for example, here's an alcohol fermentation where the pyruvates are broken down into ethanol, right? And probably the most important part of it is NADH gets reoxidized to NAD. That's absolutely essential. Probably this is the most important part of fermentation, is the fact that it oxidizes NADH back into NAD. If it didn't do that, glycolysis wouldn't continue. Because if you had a buildup of NADH and nowhere for that NADH to go, because remember ETS isn't working here. Okay? So if you had a buildup of NADH and nowhere for that NADH to go, eventually glycolysis would stop because there would be too much NADH. So really the important part of fermentation is taking that NADH and oxidizing it back into NAD so glycolysis can continue. So there's two different kinds of fermentation, an alcohol fermentation and an acid fermentation. They both do the same thing, slightly different outcomes. They take the pyruvate, they turn it into either an alcohol or into an acid. And they also oxidize NADH back into NAD. Sometimes there is a gas that's released during fermentation. Sometimes there's not gas during fermentation. So that's a little bit variable too. So production here, notice that the fermentation pathway itself does not release any ATP. It's only glycolysis that makes ATP. So you get a measly little 2 ATP per glucose along with the acid or the alcohol that was made, and sometimes the gas during fermentation. That's your overall outcome is either acid or alcohol, and then of course the gas. Now excuse me, the type of fermentation that's done does vary from organism to organism. There are lots of different possibilities. This is showing you lots of different endpoints to fermentation. Different organisms do different things. Here you have pyruvate at the beginning here. Yeast cells, we know, can take that pyruvate and turn it into ethanol. Thank you, beer, wine, etc. Right? That's a yeast alcohol fermentation. There's lots of other types of fermentation, however. If you use gluconobacter, it will take that ethanol and turn it into acetic acid or vinegar. Clostridium can take pyruvate and turn it eventually into butyric acid through its fermentation pathways. Streptobacillus and lactobacillus will take pyruvate and turn it into lactic acid. That's what starts the cheese making process, is the production of the acids from that kind of fermentation. Different possibilities here. Propionibacterium can take pyruvate and eventually turn it into propionic acid. Propionic acid is what gives Swiss cheese its particular flavor. And Swiss cheese, by the way, the reason it has holes in it is because there is gas released during this type of fermentation. That's what forms those holes. So do not memorize all of the different organisms and exactly what kind of fermentation they do. I'm using this to kind of try and give you the idea that there's lots of possible outcomes from fermentation, just depending on what that organism turns the pyruvate into during its process. Right? Sometimes it releases gas, sometimes not. Sometimes it forms an acid, sometimes it forms an alcohol, just depending on what enzymes it has to run fermentation. You can actually see fermentation in a lab by using a special type of media called carbohydrate fermentation media. This media contains sugars that can be fermented and converted into an acid, along with a pH indicator. So on the left-hand side here, this is the media before there's any bacteria in it. This is sterile. Right? Starts out as a red color. It has a particular sugar in it. Let's say it has lactose in it. Particular sugar, has a pH indicator. You put your bacteria into that tube, and if the bacteria simply grow but do not ferment, they don't release any acid, and you just see cloudiness. Right? If, however, the bacteria ferment, they release acid during that fermentation, and the acid changes the color, it's a pH indicator, to yellow. So the middle tube is showing acid production, evidence of fermentation. So it shows you that that bacteria fermented the lactose. Notice these tubes also have little upside down glass tube inside of them. That's called a Durham tube, D-U-R-H-A-M. Little upside down glass tube. If the organism releases gas during his fermentation, the gas will get trapped in that little Durham tube, and you'll see a bubble right here. So you can see not only is it fermentation, it's fermentation with gas production. That can help you to identify a bacteria. Certain organisms are known to ferment certain sugars, so you can test for fermentation of lactose or maltose or mannose or sucrose or whatever you want to, and that can help you to identify a bacteria. In fact, we'll do this during our unknowns project and use fermentation as a way to identify them. But so if we think about fermentation here and add it in, fermentation uses glycolysis as well as that extra pathway, the fermentation pathway. Turns out that fermentation uses glucose, of course, and the glucose is going to get turned into either an acid or an alcohol or a gas. The other thing here is it turns out that you must have no oxygen for fermentation to occur. Now, why is that? Turns out that all organisms that ferment can also do aerobic respiration. All organisms that can ferment also can do aerobic respiration. So if you're an organism that could do either aerobic respiration or fermentation, which one are you going to choose? Wouldn't be a hard choice, right? If you could either do aerobic resp or fermentation, of course you're going to choose aerobic resp. Why? Because you get 38 ATP per glucose instead of only 2 for fermentation. If I held out two hands, one that had $38 in it and one that had 2, and I said, which one would you like? It would take you about a half a second to choose which one you want. And the same is true for an organism. It will definitely choose to do aerobic respiration. So why in the world would it ever do fermentation? It's only going to do fermentation if it can't do aerobic resp. In other words, there's no oxygen. Right? If there's oxygen, then that organism will do aerobic resp. If there's no oxygen, then it will do fermentation. 2 is better than nothing. Right? So what I'm telling you is if you want something to ferment, you have to put it in an anaerobic environment. It's not going to choose to ferment if there's oxygen present. This is why in the beer making industry or the wine making industry, if you brew your own, you go through this process where you have to create an anaerobic environment if you want those yeast cells to produce alcohol during brewing, right? Got to go through and make it anaerobic. All right, so looking back here, we've been talking about the degradation pathways, but I want to bring up a couple of things. We've been talking about these degradation pathways, but I've always been starting with glucose as our starting molecule, a carbohydrate, a sugar. Assuming you guys aren't hummingbirds, you don't eat only sugar, you eat all sorts of things. When you eat that hamburger at lunch, yeah, you get some glucose molecules, but you also get proteins and lipids and fats and all sorts of other things in that structure. So what about all of those molecules? What about all of those molecules during that process in catabolism? Well, it turns out that all food sources eventually enter into glycolysis and TCA. All roads lead to Rome, and in fact, everything that you eat eventually gets funneled down into the same pathway of glycolysis, TCA, and then on to the ETS. For example, here's carbohydrates, right? Carbohydrates or sugars. Any of these large multi-subunit sugars will get broken down into glucose and then go through glycolysis, TCA, etc. Fats. Fats are going to go through a process called beta oxidation. And then those fats enter into the tricarboxylic acid cycle. So they go to the same place. Proteins. Proteins go through a process called deamination, and then they enter in the tricarboxylic acid cycle. Nucleotides, i.e. DNA, RNA. If you ate a burger, you had cow DNA. Right? Those nucleotides get turned into amino acids. They go through deamination and enter into the TCA. So everything eventually goes to the same place. It doesn't matter if it was a protein, a fat, a carb, or nucleotides. They all go eventually into the same process stage. Now, we have talked, if we look back at our figure here, we've talked again about degradation, catabolism. What about the anabolic pathways and anabolism? Well, it turns out that all of these pathways we've talked about can run the opposite direction too. All the TCA and glycolysis can all run in reverse in order to form macromolecules. So most of them, in fact, can go backwards to build macromolecules. So you can get energy out by breaking those molecules down through catabolism, but you can also build up the fats that your body needs, or the carbs, or the proteins that your body needs by going backwards through TCA and glycolysis. So these can go either direction in order to build your own molecules. So most are reversible to either build up large macromolecules through anabolism or break down through catabolism. All right, that brings us to the end of the metabolism lecture, and I will see you guys soon.